day, everyone. My name is Karen Picutinho, and today I will be discussing to you a particular topic in biological systematics, and these are character coding, monophyly, polyphyly, and paraphyly. Let's start with character coding. What is character? Since we are talking about biological systematics, let us focus or limit our definition in this field. Character is used to classify organisms on the basis of similarities and differences. It is also an attribute by which a member of a taxon differs. So we all know that there are many organisms that exist in our surrounding. And for sure, these organisms have similarities and differences, and that is what we need to characterize. Now, what is coding? Coding is an abstraction of observations. When we say abstractions, these are the perception or concept of observations that we've gathered from the natural phenomena that we have observed in our or we see in our surrounding. It is now also known to be a state ordering process. So what is character coding? Character coding is the link between observation and explanation. Thus, the ability or inability of alternative coding methods to reflect the evidential significance of our observations should be the primary concern in considering alternative methods of coding. Now, there are many kinds of character. One is binary character. This exists in two states, like presence or absence of trichomes in some organisms. They can be represented by the simplest form of coding, where characters are divided into positive and negative sign, or can be denoted with figures like 1 and 0. Second is multi-state character. It exists in more than two states. Such characters can be coded into number of states, like 1, 2, 3, and so on, corresponding to the range of variation. Now, there are categories of multi-state character. These are the qualitative multi-state characters and quantitative multi-state characters. First is qualitative, or it is also known to be as discrete multi-state characters. These contain three or more contrasting forms, and each form is ranked on equal footing, like flower color, which can be in any number of states, like red, white, purple, and yellow. The second is qualitative, or known as continuous multi-state characters. These represent measures of the size on a continuous scale, like length and height. There are also alternative coding methods. First is binary coding. It is the alternative way of ordering a linear multi-state character that preserves a specific hypothesis of character transformation by segregating its transformation. The second is contingent or reductive coding. This introduces non-applicable states treated as missing data when there is no logical interpretation of the character for a given taxon. This method has the benefit of allowing one to capture grouping information implicit in the presence or absence of a feature, while simultaneously capturing the grouping information implicit in transformations between trait values. Now next is Monophyly. Monophyly is a taxon which includes the most recent common ancestor of a group of organisms and all of its descendants. In this diagram, if we are going to choose G and H, then we can have a monophilic group, since descendants G and H shared one common ancestor, which is C. So therefore, this is a monophilic group. Same also with F and E and J and K. These two is or are monophilic group because F and E share one common ancestor, which is D, and J and K have also one common ancestor, which is I. Same also if we are going to take these 6 descendants, J, H, F, E, J, and K. These 6 have one common ancestor, which is A. So this is a monophilic group. Next is Polyphyly. It is a taxon which does not include the common ancestor of all the members of the taxon. Now, for us to have a polyphilic group, then we chose H and F. This can give us a polyphilic group because H has a different ancestor with F. The ancestor of H is C, while the ancestor of F is D. So this is a polyphilic group. The same also with E and J. These two have different ancestors, so it is a polyphilic group. Likewise, D and I. They have different ancestors. The ancestors of D is B, while I is A. So this is another polyphilic group. But what if we take C and D? Can we have a polyphilic group? Yes or no? Of course, the answer is no. No, because C and D share one common ancestor, which is B. Therefore, this can't produce a polyphilic group. Instead, it's a monophilic group. Next one is Paraphyly. Paraphyly is a taxon which includes a most recent common ancestor but not all of its descendants. In this diagram, for us to have a paraphilic group, then let's choose the descendants of A, which are G, H, F, and E. So if we are going to choose these descendants with the ancestors A, then we can have a paraphilic group. It is a paraphilic group because we excluded J and K, although these descendants have the same ancestor with J, H, F, and E, which is the ancestor A. But we excluded this one. We only chose the 4, so this is now a paraphilic group. And now for our research article. The title is Statistical Evaluation of Monophyly in the Broad-Nosed Weevils through Molecular Phylogenetic Analysis, Combining Mitochondrial Genome and Single Locus Sequences, the Corcolynidae, Intiminae, Cycluminae, and Hyperinae. The authors are Conrad Gillett, Christopher Lial, Alfred Vogler, and Brent Emerson. And this article was published on April 6, 2018. For the objective of the study, this study assesses the monophyly of tribes and subfamilies within diverse broad-nosed weevils through analysis of single locus sequence data for mitochondrial COX-1 and rRNL genes in combination with a backbone of complete and near-complete mitochondrial genome sequences. For the methods, first used was backbone phylogeny. The mitogenome sequences for 120 Corculionid taxa analyzed in a previous phylogeny were used in the phylogenetic reconstructions in this study, acting as a comprehensive phylogenetic framework insofar as they provided the backbone in the resulting trees. The second method is public database sequences. Automated extraction of sequence data from GenBank was achieved through the use of a series of Perl scripts originally developed as part of a custom-built bioinformatics pipeline for analyzing public database sequence data. Third is Monophyly constraints. In order to test whether Monophyly or any of the subfamilies Intiminae, Cyclaminae, and Hyperinae, and any of the tribes within the subfamily Intiminae were consistent with a combined dataset. Fourth is phylogenetic analysis. The dataset was analyzed and partitioned by gene. And lastly, the statistical hypothesis testing. The per-site log likelihood was computed for each of the unconstrained and 20 constrained trees in RAXML using the FG algorithm and written to a tree puzzle formatted file. For the results, moderate and high bootstrap support was consistent with two Intiman tribes, being each recovered as monophyletic in an unconstrained analysis. Furthermore, one genus of cyclamin weevils called Aphela is recovered outside the clade of broad-nosed weevils, although its taxonomic placement remains uncertain. Conclusion. It is apparent that the present approach may be hampered by limited taxon sampling in the backbone dataset, rendering it difficult for divergent taxa to robustly match to their closest lineages. However, with improved taxon sampling of the metagenome tree, the general approach can be a useful taxonomic tool for weevils. And these are my references. So that's all. I hope you enjoy. I hope you learn.